And so Dr. Zakee Sabree joins us here today from Ohio State University, where his lab has done an incredible model system using germ-free cockroaches to act how microbes mediate host development and nutrition. He also works with other types of insects, so that genome shrinkage and obligate insects in this environment. So he received his bachelor's in biology from Howard University in Washington, D.C. And from there went on to complete his PhD at the University of Wisconsin-Madison in the lab of Joe Anderson. And then he worked as a postdoc at the University of Oregon and, University of Arizona and Yale University and Moran, where he published some of the early papers characterizing the gut. So without further ado.
Great. All right. Thank you. Okay, so just I really do appreciate the invitation to come out and share my work, especially like after the pandemic. Giving talks over Zoom was not that satisfying compared to actually being able to come to places and visit and sit in rooms. So it's a real pleasure to come out here. I tried to match that green with Oregon green. It just turned into a party. All right. Okay, so I don't know if the people on Zoom can hear me. Do you know? Okay.
Yes.
Okay. All right. Great. Cool. And so I assume everyone in the back, if you can't hear me, just let me know and I'll just talk louder. So yes, I'm going to talk a little bit about the work that we've been doing in our lab for the last few years. So I always kind of start with the people. These are the people that do a lot of the work in my lab, graduate students, undergrads, and postdocs that have all contributed to this work, as well as people that have funded a lot of our research. And so without their contributions, this wouldn't be possible. So my lab is really interested in symbioses. I've been using this slide literally since I got my first job. And it's in part because I think that it really does kind of capture in a very simple way the kind of breadth of known kind of interactions between microbes. And so my lab from the very beginning was really interested in trying to uncover the evidence of interspecies interactions. And so I accept a pretty broad definition of symbioses. I think when I got started, I think there was a little bit more reticence to acknowledging that pathogenic interactions could be symbioses. But I think that's changed quite a bit, in part due to several of the people in this room, as well as other folks in sort of the microbiome science space. So I think this is also maybe somewhat obvious to a lot of people here, but I've given talks in departments where I have to kind of remind people that metazoans evolved in a microbial world. That's not always obvious to a lot of people, and what are the implications of that? And so by showing basically, how long bacteria have been here, and then how everything else kind of came up, they had done a lot of work and have been integral in shaping the evolutionary trajectories of lots of metazoans. And so this is always fun, showing how humans are kind of like right here and have just shown up, yet their impact on our ecosystems is quite outsized. And so we work primarily with and in insects, because insects are really, really fascinating because they populate, it's at the very least populate land masses, as well as many of the waters as well. And a part of what has allowed them to be so successful in populating all these very different types of environments, we think is in part due to their associations with microbes. And so in many ways, microbes enable insects and invertebrates to be able to kind of explore evolutionary space at a much faster rate than having to sort of evolve some of these functions de novo. And so I have a couple of examples up here, but that kind of takes me into working in insects, because a lot of these relationships that I have up here involve microbes that are living within tissues of these various insects. So everything from living inside their digestive tracts to inhabiting specific cells within these insects. And so we have, like, not only do we have microbes that are maintaining these sort of binary relationships with their insects, so it's just like one insect, one bacterial species, but you can also have assemblages of bacteria that are living within their insect host, and even sort of associated with other microbes like this protus that has all of these spirochetes and members of the Bacteroides decorated on the outsides of them that live inside of termites and sort of assist with fixing atmospheric nitrogen and processing complex carbohydrates. So insects are a really wonderful kind of clade to work with because they have a wide diversity of relationships with bacteria, and a lot of those relationships are beneficial. Also, no one cares when you kill insects, except entomologists maybe. So, so we can do a lot of experiments with insects that would be maybe really expensive or really hard to do in vertebrates, and should be prohibited in humans. All right, so that's sort of the reason, another kind of added value. All right, so I've done work that has looked at sort of obligate endosymbiosis. So these are bacteria that sort of live restricted, incarcerated within their animal host tissues. They're typically vertically transmitted. And then oftentimes it's just one or maybe two species of bacteria, and they oftentimes provide some very specific services for their host. And so I did a lot of this work in cockroaches with Bladybacterium, and then also some other insect species as well. And this particular sort of syndrome or symbiosis syndrome presents some really kind of fascinating kind of evolutionary outcomes. And so I won't be talking very much about that today, but this is where a lot of my research began. And I kind of boldly in the middle of my career said, I don't want to do this anymore. Let's go do something else, which I think was quite the gamble. And I got more interested in, well, not more interested, but I also got interested in looking at gut microbiomes. And so gut microbiomes is on the lips of my mom, which means it's everywhere. And so these can sort of be comprised of lots of different species or even just a few. People are characterizing their ability to participate in diet processing and protecting the host from pathogens, a variety of different potential services, and they can be transmitted vertically, it can be transmitted horizontally, acquired from the environment. So there's a lot of space in here for exploration, and people are doing some really amazing work being able to sort of assign functions to microbiomes. So I got also interested in this because in, I don't know, probably it was like earlier part of the aughts, I was reviewing papers and and people were doing a lot of microbiome work and assigning sort of functions to these microbiomes without a whole lot of evidence. In the sense that, whereas it's like, we see this microbiome, we characterize all these different taxa that are there, and based on what we know about these taxa, we infer that they're capable of doing these things. But many times it kind of would stop there because there just weren't tools to be able to test these hypotheses. So it wasn't that people were doing bad science, it's just, it may have been hard for them to actually sort of test those hypotheses. So that's what got me interested in developing the cockroach system, to begin to sort of add to the number of tools that were available to investigators to begin to test these hypotheses. So I hope to convince you to some degree of why this is a valuable system and kind of show you some of the work that we've done. So this was a really nice review that came out a few years ago, kind of sort of showing how there's lots of different animals that have these different types of digestive systems inhabited by microbes that are performing a variety of different services. everything from diet processing to nutrient provisioning, so directly providing nutrients to the host, like short chain fatty acids, vitamins, etc. That these microbes are integrated into the development of their host as well. So sort of protecting any pathogens either through sort of actively competing with them or providing a barrier to sort of not allow for pathogens to be able to inhabit them. And so, oops, what happened? Oh yeah, so basically, we can have these microbiomes that are oftentimes comprised of, either just a few different species, or they can be quite complex, and complex meaning comprised of a lot of different species, which we'll get to in a moment here. But what is kind of interesting and something that we've been really kind of obsessed with for the last few years is in these complex microbiomes, what of those members are required for supporting their host? So do you need all of them? Let's see, do I have a pointer? Yeah, here we go. Do you need all of these taxa to be able to support the host? Because we're talking about sort of a host beneficial microbiome. Or is it possible that there are, say, subsets of this microbiome that are able to also support the host? And so are these subsets overlapping, or are they distinct? And so this is in part because through a lot of the metagenomic work that's been done, it's been shown that there can be a fair bit of functional redundancy across lineages that comprise these microbiomes. And so therefore, it's not unreasonable to think that you could have some loss of some of these species and still be able to provide all the resources to the host that's needed. But systematically testing this, especially in really complex microbiomes, has been somewhat challenging. And so we see versions of this type of work with reduced microbiomes, so these nautobionts that are sort of polyassociated and looking at various features. But we are also interested in trying to see how to disentangle or to even disrupt these complex microbiomes as a way to figure out what lineages are essential for normal host growth and development. All right, so just a little quick history. So like germ-free animals have been around for some time. People have been working with them for over 100 years. And germ-free mice and rodents are really well known. They're really useful because of the easy translation to humans in some ways. But they're also very expensive. You require a lot of resources to be able to maintain them. And so germ-free invertebrates have kind of been ascendant as a result. And for a lot of reasons that I had mentioned earlier, just about working with invertebrates in general, but also there can be a fair amount of sort of analogous, at the very least, genes between vertebrates and invertebrates that makes them really valuable. I have Drosophila up here because they're pretty ideal, because there's a lot of genetic tools and already sort of genetic lines available for asking really great questions in them. But now that we have things like CRISPR and deeper CRISPR available, now that it'll be possible to potentially do some similar types of work in emergent model invertebrates as well. What's also ideal about invertebrates, including Drosophila, is that they're amenable to germ-free lifestyles, and so you can rear them germ-free. They have some clear phenotypes, and then you can sort of return microbes to see if that kind of relieves some of those issues as a way to sort of assign function. So while invertebrates are really, really nice, oftentimes because they're pretty cheap as well. So like germ-free mice are about $300 a pop, maybe even more than that now. Germ-free insects can be about $1. And actually for us, germ-free cockroaches are about 30 cents each. And so clearly there's a cost scale there that's really nice. But besides that, we find that in some invertebrates like Drosophila, honeybees, and like C. elegans even, the microbiomes are pretty simple relative to what we might find in humans and even in mice. And so given that, as well as the sort of the somewhat different anatomy, being able to be analogous to what you would find in mammals can be somewhat challenging. And so this is just kind of a quick breakdown even of the, obviously for reasons when trying to think about how microbiomes, especially complex microbiomes, are playing a role in host growth and development and evolution. So what was also nice about Paraplaneta is that, they're omnivorous. And so if you've ever had to take out the trash, you've probably seen them, right? And so they're amenable to our diets, and therefore we can ask questions that are linked to how diet is impacting the host through the microbiome pretty easily without it being too far of a stretch. Also, as I said earlier, they maintain a complex microbiome, and we found that many of the bacteria that comprise the cockroach microbiome are actually culturable. And we've been successful in doing that, and I'll talk a little bit about that. And what's nice is that many of those culturable bacteria are congeneric with those that you find within humans and mammals that are also omnivorous. All right. So it's really easy actually to make cockroaches germ-free. We don't use antibiotics, right? So we don't have those issues of sort of hitting them with all these drugs to be able to get them to be germ-free. Because they lay these egg cases, which kind of looks a little bit like a Balenciaga bag, frankly. But all you have to do is just basically surface treat it with bleach and soap and some enzymes, and that's it. And then we place them into these plastic tubes. It's just some rubber tubing that were used, I think, for the Bunsen burners in the lab. And then we provide filtered air, gamma radiated food, and we provide water in the form of low concentration agar plug. And then that's it. And they will develop germ-free until sort of a later juvenile stage where they then plateau. And that's actually kind of an area of interest for ours as to why is it that our insects, or germ-free reared insects, kind of plateau in their development and don't make it to adulthood. That's a side piece. But we do know that if we rear them germ-free and then we conventionalize them with frass from conventionally reared insects, it resolves all those problems. We also have developed a approach for making monoassociated and polyassociated bionts as well. And we have done work where we've cultivated, I think at this point we have about 115 isolates from our cockroach now, and we've been able to sort of reassociate them with our azenic reared insects successfully, and they colonize. All right. Oh, and so the equipment that's required for being able to maintain, say, a colony of about 300 azenic reared cockroaches is about the size of a Nike. Y'all are familiar with Nike, right? Like a Nike shoebox, right? You can keep that many in there. And so to be able to have sort of large populations so you can get the kind of statistical robustness for your treatments is possible with a fairly low footprint, right? So we have growth chambers that are about the size of a typical refrigerator that can have, 15 treatments each with about, 100 to 300 individuals in there. So it's really nice in that way. All right, so I would not have gone down this path unless there were some very clear phenotypes of azenic or germ-free rearing that were then resolved by returning their gut microbiomes. And so here I kind of highlight one of them. So this is the digestive tract from a germ-free cockroach, 5th instar. So that's sort of a later juvenile stage. And then at the top, you'll find the same tissues in a normally reared. So when we observe this, and we observe this consistently, that the germ-free insect digestive tracts are highly differentiated between the midgut and the hindgut, which sort of have distinct functions and also have distinct microbial communities associated with them. We also find that the musculature that is involved in sort of the peristaltic activity is diminished. So just a variety of different very clear phenotypes. And as far as the musculature, you can see that more through these cross sections, which I have kind of arrayed below and above. And so when you actually look at the luminal surface for our germ-free insects, you have a lot less surface area. So a lot of the crypts that you would expect and that we see in the normally reared cockroaches, you don't find them in the germ-free. And this is similar to what we actually see in mice as well. So it was really nice. And actually, in some of the photos you showed me today, Karen, it was very similar to what we see also in the in fish too. So that was really nice, and it was actually really nice to see that from a sort of a comparative biological point of view, that microbes are impacting digestive tract growth and development consistently, even across sort of like deep evolutionary divergences. We also, and so, I mean, this is great and fine, but you have to do your dissections to see this. So what was really nice is that in body size for age-matched insects, we saw that the bodies were quite a bit smaller for our germ-free reared insects compared to wild type. And so this was nice because this is something we can have our undergraduates do, right? So we have them involved in doing research, and literally they're just coming in and looking in the the bins and just looking at who's too small, who's too big. They count that up. There we go. All right. So this model system is really great because it can engage students and researchers at different stages of their own development, right, and their own abilities. And so we like that because we can get undergrads doing this and counting hundreds of cockroaches when I don't want to do it. So also, in addition to the body size being smaller for age-matched insects, we also found that development time was about 30% longer. That's what we have over here. So like in the blue, we have our sort of normally reared. In the red, we have our germ-free or azenic reared. And what we generally found was that for each instar, so cockroaches are hemimetabolists, and so they molt each time they get ready to, when they grow, right? So the molting time, the time between moltings for our germ-free insects was about 30% longer than it was for our normally reared insects. And so that would, accumulate over time to being, quite a bit longer than it would be. So that was really nice. And we could resolve that by just giving them poop from normally reared cockroaches, which was really nice. We also found that the digestive tract lengths were different. So like the hindguts for our germ-free insects were shorter and were also underdeveloped relative to what we saw in our conventionally reared insects. The midguts, there was a trend of that, but there was a lot of variation there. So we couldn't say for sure that they were significantly different and just different. You know, say significantly, just different. But we decided to drill down into that a little bit, and I'll talk about that in the next slide. So based on some work that's been done in Drosophila showing that there is in some ways a regionality of microbes along the length of the gut, suggested that there were parts of the gut that were, being colonized by bacteria, more readily, more frequently than, other parts of the gut. Namely, the hindgut tended to be colonized more readily and more consistently than the midgut. And so we wanted to kind of see if there was a similar trend in our cockroaches where we have these really distinct mid and hindgut regions. I mean, they look different, and based on all the physiology that's been done in cockroaches suggests that they have very different roles. The midgut being about dietary nutrient uptake, and then the hindgut being kind of more where you have some like processing of some of the more recalcitrant parts of the diet, by the bacteria that reside there that then produce short chain fatty acids that kind of feed those tissues, and maybe a little bit of like, elimination of some waste material as well. But given those two different regions and the functions associated with them, we thought that perhaps we might see different impacts of the microbiome on the growth and development of these tissues. So what we did is we regionalized or define these subregions of the digestive tract. So an anterior and a posterior, mid and hindgut. And then we did just a lot of histology to investigate if the microbial status impacted various features of the digestive tract. So we looked at everything from the surface area of the lumen. So that would allow us to be able to kind of capture the depth of those crypts. We also looked at just overall thickness of the tissues and then the outer circumference as well. And so what we found when we had our sort of conventionally normally reared cockroaches, so when we say normal lab reared, we took germ-free cockroaches and then gave them poop from sort of our non-sterile culture that we keep. And so we had germ-free, and then we also had a third treatment, which was we gave them a single exposure to a normal gut microbiome and then prevented them from getting any more after that. And I'll talk about that briefly, but what we saw was that in the anterior midgut, we saw less of an impact of microbial status on those tissues. But the further back we went through the gut, we started seeing that the microbial presence had an increased impact as we kind of got to the posterior region of the hindgut, which is where we saw, primarily in the hindgut in general, we saw the most impact of microbial status. And so that kind of suggested to us, given that the hindgut is where we typically find sort of, the highest abundance of bacteria, and also that these tissues are likely where you have, microbes that are involved in sort of, diet processing, that maybe they would be more impacted by the presence or the microbial status. Whereas in the midgut, we were thinking that this might actually be sort of a region of competition between the host and its microbiome, because this is where you're going to have the dietary nutrients that are probably going to be the most easily assimilated. Why do we say that? Because this is just kind of recently after sort of mastication and chewing, the releasing of these sort of, simple sugars, proteins, fats, things like that, that those are now, in the gut. And so the microbes that are there could likely be taking those up and utilizing them, as well as the host. And so we came up with this model of this kind of, competition collaboration between the host and the microbiome based on the region of the gut that you're at. And so I think this isn't a novel idea, but it kind of goes against a general kind of idea that there's this sort of kumbaya between the host and its microbiome along the entire length of it. And we don't think that that's entirely the case. And so what we did to kind of look at this a little bit more is we did some transcriptome analyses, focusing primarily on innate immune gene activity. And so innate immunity, for some of the, I don't know, mammal biologists in here, insects have immune systems, if you didn't know that. So I was at Yale many, many years ago as a postdoc, and I was talking about this, and this very famous person said, insects don't have immune systems. And I about died because I was like, wait a minute, what? I've read this in several books. But anyway, so insects have immune systems. And so we wanted to see whether or not there was also a regional activity of the immune system, okay? And so what we did is we took our germ-free, we had another treatment, our non-abiotic, which I can talk about a little bit later, and our conventional reared insects. And so these are PCOAs, and they're backwards. That's not important. I turned them that way because I wanted to kind of orient them in a midgut and hindgut sort of way. So we focused in primarily on toll and IMD. So these are two major parts of the innate immune system involved in sort of sensing microbes that are present and then responding by either sort of damping down the immune response or by producing various antimicrobial peptides and such that could kind of kill off, parts of the microbiome. And so what we did is looked at midguts and hindguts for an expression of all the genes comprising the toll pathway and the IMD pathway. And what we saw was that between the midgut and the hindgut, it looked as if in the midgut, there was a marked effect of microbial status on expression of innate immune genes relative to what we saw in the hindgut between toll as well as IMD. Much more the case in toll than we see in IMD, but it's that spread that I'm trying to highlight here. And so this was like, okay, that's kind of nice, but what about in terms of this competition aspect? Well, what we found was that atacin, which is a antimicrobial peptide that is active against different types of bacteria, that atacin and relish, which is a regulator for other AMPs, that these tended to be downregulated when you had just, these tend to be downregulated in our germ-free insects, but in the normally reared, they were active, right? We didn't see that same kind of difference of expression of atacin and relish in the hindgut, right? So that gives us some further support that in the midgut tissues, when you have your typical microbial status, that these are genes, these antimicrobial peptides and innate immune system may be on, and they're being used to compete potentially with the host for these nutrients. Also, these killing of bacteria actually generates available nutrients for the host too, right? So it's not just simply, so innate immunity is not just for protection, it can also be for digestion, all right? So we're exploring this now further and doing some work to actually do some qPCR for a number of different antimicrobial peptides as well as AMP regulators to see how widespread this is. But we think that this sort of gradient of relationship is an exciting area and in a way to begin to sort of think about how the microbiome and the host are in a much more dynamic relationship than maybe how we maybe previously thought about it. All right, so I'm going to just switch gears just a little bit here and kind of talk about how when using these microbiomes or thinking about these complex microbiomes, how we can sort of, based on the taxonomy that we observed there and then even doing some genome sequencing, how do these inferred functions suggest something about their importance to the host? All right, so I said earlier that I showed you those bar charts that were hard to read. And here we kind of break them out a little bit. So we find that within Paraplane americana, Bacteroides, Bacteroid DTs, Firmicutes, Proteobacteria, these are common. And you find that they're also common within vertebrates, mammals, humans. And so a lot of the same lineages that predominate in those, like members of the Bacteroidaceae, Ruminocaucaceae, that are abundant within mammals are also abundant here. And so we were able to, use this to develop cultivation approaches to begin to sort of get many of these bacteria into culture. So we have isolates for, as I said earlier, about 110 aerobic and anaerobic bacteria that are commensals to cockroaches or to Paraplaneta. And we started doing some phylogenies with them to kind of get a sense of what's the possibility that these are maybe tightly associated with the host, or do they just pick them up like rummaging around in the sewers or something? And it looks like many of these bacteria are typically associated with this host insect. And we say that in part because we consistently see cladogenesis for our isolates. And so that's what those red boxes basically showing, all these new clades of bacteria that we are finding within Paraplaneta. And so it's seductive to think that maybe bacteria are, say, important for the host and are maybe, I don't know, I think some of the very positivists of us might even say that these are mutualist or something. I would argue that there's an alternative hypothesis that I'm not rejecting mutualism. We don't have the evidence for it, but a really good hypothesis when it's to consider the possibility that many of these lineages may be, speciated through allopatry. So it just got stuck in the host. And as, the host basically speciated, they just, caught a ride along with them. And I think it's especially the case for oxygen sensitive bacteria, that their migration ability is really limited. And so as a result, drift can have its way with these bacteria. And so they end up sort of speciating because they maybe rejoin the larger population for recombination and change. So that's one way to kind of think about these sort of new lineages that kind of emerge. And let me see, I'll just fast forward here a little bit. And so I was just showing you some members of the Bacteroidetes that did that, but even for some of our Firmicutes that we've been able to cultivate, we still see not only that they form new clades, but also they even kind of have long branches. And that further suggests that, that they're kind of speciating in their host, and it could be due to some drift effects that we're observing these extended branch lengths relative to other bacteria that are associated primarily, at least with these, associated with mammals. So to kind of come back to the bit about like inferred function or even tested function suggesting something about importance to the host, a lot of these Bacteroides that we've been able to cultivate can participate in various parts of complex polysaccharide degradation. So everything from polysaccharides to even sort of animal-produced glycans. We have just within a subset of these capable to break down all of these major complex polysaccharides. And so look at this and we're like, oh man, they must be super important for the host. I mean, all we got to do now is just put them back in the host. The host will be and feed the host its normal diet. They'll be happy. They'll develop normally. Science paper. So we did that. We also included a couple of Firmicutes that we had cultivated as well that also had the ability to be able to break down lots of complex polysaccharides, including cellulose, and also appear to have other functions that would be useful for the host, like provisioning vitamins and such. So we did a polymicrobial associate, or basically we cultivated them all individually, mixed them together 1-to-1, and then put them back into our azenic reared insects. And what we saw was kind of nice. All right, so when we compared the expression of several carbohydrate degrading genes in our germ-free versus our non-abiotic, we saw that in germ-free insects, they were upregulating a number of their endogenous carbohydrate degradation genes, where we returned these bacteria, they would go down in expression. So it might be a little hard to see that. So this is germ-free level of expression compared to non-abiotic. So they're upregulated. When we compare the germ-free to wild type, we see kind of a similar pattern, right? So when we compare expression of these genes between non-abiotic and wild type, it was basically this, we don't see any differential of these carbohydrate degrading enzymes. So we think that, wow, that's great. It looks like these bacteria are able to, at the very least, signal to the host that expression of these genes or upregulation of these genes is, not necessary. So we're like, well, then these cockroaches must be able to then grow normally and just be just fine. That was absolutely not the case. What we found was that our germ-free and non-abiotic insects were like size, so they're both smaller than wild type. They're not this big, but as they're on the screen. They were definitely smaller than the wild type. And then when we looked at development time, when we looked at other aspects of body length and mass, when we looked at the gut lengths, they were all germ-free and non-abiotic were identical. So what that then suggested to us is that it was a bit more complicated than just, even if you have these abilities to be able to help with dietary processing and even change expression of these endogenous carbohydrate degrading genes, there's still maybe more that's going on. And we also could have just picked the wrong bacteria. And so when we kind of explored this further, we found that in general, in our germ-free as well as in our non-abiotic insects, we found that their carbohydrate digestion, sort of all the genes involved in that, it looked as if, like in that as well as in fatty acid catabolism, it looked as if they exhibited this starvation approach because they were upregulating all of its own endogenous enzymes that could help with being able to scavenge whatever carbohydrates that are there, right? So this was, like I said, it was a bit disheartening because we thought that this small subset of commensals would be capable of complementing normal growth and development, but that was not the case. And we also saw that in genes involved in regulating sort of growth and development, like insulin signaling, we saw that they were downregulated in both germ-free as well as in the non-abiotic insects. And so that we think, helps us to explain this, the smaller body size, the prolonged development time, as well as the digestive systems that were like a total mess compared to the wild type. So this is kind of useful when thinking about when we put our results within the context of work in honeybees, as well as in Drosophila, where you could return one or even a few members of these smaller microbiomes to their host, and they were not only able to sort of relieve the expression of, in this case, insulin signaling genes, relieve what was observed in the germ-free. Just by giving back one or a few bacteria, you were able to resolve that. They were also able to resolve some of the other sort of growth defects and developmental defects that were observed in these insects. So what this suggested then to us was that in some ways our somewhat arbitrary, and I say somewhat arbitrary because we looked at the genomes and we were like, they can do great things for their diet. They must be important. But these somewhat arbitrary assemblages were not capable of relieving the dysregulation that was observed in germ-free. And more importantly, we're not able to recover wild-type growth phenotypes in our insect. And so that's led us to this next investigation, which is to be less arbitrary and maybe a little bit more random. And what do I mean by that? So as I said earlier, you have this sort of complex microbiome with lots of different species. We don't know which of these are entirely necessary for like normal growth. And so we don't know if there are, what subsets there are. What we can do, because cockroaches are naturally coprophagic, we can take their frass and we can actually just do dilutions and then return those dilutions of the frass to our germ-free rear effects. And then basically we're going to look for normal and abnormal growth phenotypes. And then we can go back and then we can profile those microbial communities, see, who's comprised. While we think that the lower probably work great, like many of them, if not all of them will work, we think as we really begin to sort of reduce species diversity and ostensibly probably functional diversity, that we'll have fewer and fewer cohorts that will do well. But because, again, 3 cents a pop, we can do lots of these. We plan to do lots of these. So that's sort of the future directions for that work. All right, and so I do want to kind of briefly talk about sort of the other work that we're doing on this sort of genome evolution component of gut microbiome constituents. So we're really kind of interested in exploring further how the impact of these life history traits, like being oxygen sensitive, driving what looks like a signal of phylosymbiosis or sort of cophylogeny, right? And so we're in the process now of finishing up sequencing another batch of about 100 different isolates from our cockroaches. And we plan to do basically a bunch of compare, sort of explore as perhaps allopatric speciation maybe better explain what we're observing in terms of sort of cladogenesis for host-associated bacteria than perhaps just gene content or what we maybe think is, nutrient provisioning or support. So that's another project we have going on in the lab. And let's see, how are we doing on time? Well, I guess are we done? Are we done? Is it 1:15? Okay, well, I don't want to talk till 1:14. All right, so just to kind of sum it all up. So basically, we think that this is a really good model because you have a complex microbiome comprised of a lot of lineages that we think are that are culturable, that share genera with lineages that are found in lots of other animals. They can eat a lot of the same things if you're interested in sort of looking at how diet can impact the microbiome and host growth and development. It also looks like that there needs to be, at the very least, just genomes of the bacteria that are in the microbiome may not be sufficient for suggest if they're going to be capable of supporting the host completely. But that's there's an ellipsis at the end of it, because I think that we were looking primarily at a dietary feature, that there could be other aspects of the genome that could suggest that. And so this is what we're exploring further. But they're really, really nice model system. You can rear them germ-free without using antibiotics. It's very cheap. It's an opportunity to ask a lot of questions about sort of a complex microbiome in an animal without it being too artificial. All right. Oh, last thing, pay your postdocs. I'm also the faculty director for the Office of Postdoctoral Affairs. So I always say this, pay your postdocs, right? Salaries for postdocs have not changed, even though the salaries for PhDs outside of academia continue to go up. And if you can afford it, please put money in your grants for helping your postdocs get to you, okay? So this is a program we started at OSU. I got a big grant from Kellogg Foundation to do this, but I also just tell faculty like NSF, NIH, unless it's maybe one of these specific training grants, you can always write this kind of money in. So definitely, help your postdocs get to you. It means a lot. $5,000 doesn't mean as much to faculty, but it means a lot to our postdocs. So, all right, that's it. Thank you. Karen.
Essentially colonize sort of by a third or three cockroaches with humans.
Absolutely. Yep. Oh, yeah. So can we force cockroaches to eat human poop? Yes. So we haven't done it. There's a group in Germany that's about 5 used mouse microbiomes similarly. And then you had this basically really nice host filtering of the lineages to basically begin to assemble what looks like sort of the typical commensal microbiome. We are, as we talked about earlier, looking to do that. Yes. Thank you. So different microbes between the different regions of the guts. We do have that there are distinct what is it? There's a profiling, but it's more about around the relative abundances of the bacteria that are there. But if you can find a lot of the same things in the hindgut that you would find in the midgut, right? It's just they're less abundant, and it kind of switches a little bit between the mid and the hindgut. Yeah. Well, so I'm arguing. Okay, ask the question. What's the question again? So so the question is, there was some, I maybe didn't effectively communicate what I meant in terms of if there's allopatric speciation and co-speciation of these bacteria that are present within the guts. Is that right? Right. Okay, so are they co-speciating? Is okay, yeah. So I do think that these bacteria are co-speciating in the sense that they are, like, as the host speciates, I think that's also happening with these bacteria, but it may not be because the bacteria are providing any services necessarily for the host. It may not be a mutualism, right? There may not be any kind of like active selection for those particular bacteria, right? It could be just, I mean, not to say that that isn't happening. I guess I'm kind of pumping the brakes on a mutualist, interpretation in the absence of like evidence to show that. What I'm trying to say is that there are other ways we could explain why there's co-speciation, that it could be as a result of just allopatric species, like they're just stuck inside the host and drift is impacting them in such a way that they are just kind of diverging away from the larger population or populations found in other species or in the environment. And it's because they're stuck. Yep. Thank you. Man, so why do human and cockroach gut microbiomes look so similar? Oh man, that's a hard question to answer. I don't know why. So, I mean, so we don't know. I don't know. There are certainly dietary similarities. There's even like sort of like the oxygen profile of the digestive systems are similar. But I don't know. I don't know why they're so similar to one another. Yeah, and another problem with this is that, you know, cockroaches are so ubiquitous, right? And we just don't have good sort of, I mean, we don't have any like pre-human specimens where we can look to see. But I mean, it looks like, I think, you know, an omnivorous diet, I think, has a huge impact on this. Because if you look at, say, some of the more herbivorous primates, their microbiomes look quite different. So there's that. I mean, yeah, I don't want to say any further than that because I just don't know. This is gonna be on YouTube, man. I don't want to say anything crazy. Hey, aren't you a scientist? No, that's fine. That's fine. Because then they're like, why did he pause it? Yes. Mhm. Nope. Well, actually, yeah, okay, so is it because of, so does the hemimetabolous, holometabolous difference have a big impact on microbial community composition and then their impact on sort of growth and development? Is that right? Does that work? Okay, this is for the people in space, you know? All right, so cockroaches actually do purge their fore and hindgut through each molting, right? So this is like a natural perturbation that we are exploiting for another experiment now, that allows us to be able to sort of look at, like, you know, how the microbiome sort of persist. So is the midgut just seeding, you know, forward and back, you know, after each molting, or is it just consuming, which they happily do? They'll just consume frass nearby, right? Because they're not all molting at the same time, right? And it's just, you know, there's just poop there too. So so they're either kind of reseeding from consuming that, or there's this sort of like the midgut is basically providing the microbes for the hindgut. And so what's cool about this is that we can actually interfere with that, right? So we can go in and we can just collect all the exuvia, collect the frass following a molting, and then look at how the microbiome either stays the same, or if we see a drop in sort of species and functional diversity. And we have some evidence that suggests that that's actually happening. So if you prevent them from, you know, feeding on frass from, you know, nestmates or whatever, that they actually begin to kind of, you know, there's a decline in microbial diversity, and it does appear that their digestive system doesn't develop as normally as insects that have an opportunity to constantly sort of refresh and re-up, right? So it doesn't look like it's the midgut microbiome post molting is sufficient to recreate the microbiome for the hindgut and the foregut. So I think that, yeah, with like these holometabolous insects, yeah, because yeah, the microbiome can be quite different between larval and adult stages, and they're oftentimes like sort of acquiring that through, you know, like vertically and horizontally as well. And it's always happening in the colony, right? So that's a way for them to sort of reacquire that, you know, after they emerge as adults. So, and they also have a less diverse microbiome too. They've evolved with these sort of, you know, with a dozen or so lineages versus say hundreds. And, you know, based on some of the work like of Nancy Moran and some other folks whose names escape me because I'm nervous, basically, they've shown that, you know, there's some kind of, you know, niche partitioning of the functions. And so that has like been a really powerful kind of selection for those lineages that are there. So, so yeah, I think that the type of development does have an impact, and then size and the amount of sort of redundancy between lineages that probably has some impact on that too. Yes. Thank you.
So, behavioral differences between wild-type populations, for example, I'm thinking something like and if you do, with those behaviors could also cause some of the
Yeah, so are there behavioral differences between germ-free and normally reared cockroaches? And could some of that impact sort of, their, like, are they less hungry and so therefore their body sizes are smaller? Yeah. Yeah, no, I think that's totally valid. We don't observe a change in feeding behavior. We have not spent a lot of time looking at behavior between our azenic and our conventionally reared, or even our non-abiotic reared. But that is something that we are interested in looking at. One feature of our germ-free cockroaches that I'm curious about is sort of light responsiveness between the azenic and the sort of conventionally reared, because like there was some work done with like mice where they would take a germ-free mouse and put it on the floor in the dark and cut on the lights. They would just kind of sit there and look, which is a maladaptive behavior, right? Because they're just a nugget for something to eat. But then they could feed these germ-free mice frass from like just sort of a non-sterile reared community, and in a few weeks they would do the same thing with that now conventionalized mouse, and they would do the lights and run to a corner. So that kind of very sort of adaptive behavior that appears to be linked to the microbiome is something that we would like to try to do something similar with ours to see if there are some behavioral differences, but we haven't done that yet. Thank you very much.